they're microscopic. And so the question is, why is that? And how small can they go? And how big can they be? Are there limits? Are there upper and lower limits on cell size? And of course, the answer to that question is yes, there are limits. So we look at lower limits. What would limit how small a cell could be? Well, there has to be enough space to accommodate the DNA and proteins, et cetera, that are needed in order to maintain homeostasis in that cell. That being said, look at that organism that's up there, mycoplasma. Who recalls what myco means? What root that is? That's myo. That's close. Yeah, it's fungus. Very good. We used to think this was a fungus because it was so slow growing. It's not. It's misnamed. It's actually more bacterium, although it breaks some of our bacterial rules. But look at the size of that mycoplasma. What is that symbol? Does anybody know? What is a micrometer? A micrometer is a thousandth of a millimeter. I mean, think about that. It's a millionth of a meter, but it's a thousandth of a millimeter. How can a cell be a thousandth of a millimeter? That is like mind-boggling to me. But that's what a micrometer is, 1 times 10 to the minus 6 meters. So that's about as small as we can see and still have room for the stuff that needs to be inside of the cell. Well, how is it that the viruses can be so tiny?
Because they're only DNA.
Yeah, because they don't have all that stuff, right? They use the cell for the rest of that stuff, so it allows them to be a much smaller size. So what about upper limits? Are there upper limits on how big cells can be? Well, obviously there are, because cells are, for the most part, microscopic. So there are a couple of things that are limiting factors on cell size. The fact that we grow by making more cells, not by making bigger cells. But one of those factors, and one that's very important for you to understand for your short FRQ, as well as the lab that we're going to be doing next week, is this whole idea of surface area to volume. And that ratio is critically important to that cell being able to maintain homeostasis. So what we know mathematically, and that chart up there shows it to you, is as things grow in size, that surface area to volume ratio gets smaller. It decreases. So when we look at a cube, for example, here we have an illustration of a cube that is a centimeter on each side versus one that's 5 centimeters on each side. We can see what happens to that surface area to volume ratio. That surface area does not keep up, so to speak, with that volume. It does not increase at the same rate. And that poses problems for a cell. Why would that be a bad thing for the cell? Well, think about what would be represented by surface area in a cell. What part of the cell would be the surface area? The cell membrane, right? Okay, the cell membrane, which is very important because it regulates, determines how things come and go from that cell. Whereas the volume would be represented by the cytoplasm, right? All the stuff that's inside. So the idea here is if I think about surface area, cell membrane, as being the roads in and out versus the volume, the cytoplasm, as being the population. If the population is growing at a faster rate than the avenues in and out, what's going to be the result of that? What happens if my roads don't keep up with my population? Yeah, traffic jam, gridlock, overcrowding, right? And that's what happens with the cell. As that cell grows and its needs are greater and greater, there is not enough space for things to move in and out of the cell fast enough. This is not, and I cannot emphasize this enough, an issue where the membrane ultimately is going to burst or explode. Okay, that's not what we're saying. The membrane will always be there. There just won't be enough space in that membrane to keep up with what needs to happen in the cell. Okay, so that's a very much a limiting factor. The other limiting factor, which has to do with that indirectly, is the whole idea of diffusion. Okay, many of the materials that the cell needs come and go either through diffusion or facilitated diffusion. Diffusion happens very slowly. It's efficient because it's passive transport, meaning it does not require ATP, but it happens very slowly. So how does a cell overcome that speed? By having the distance be very short, right? I read a statistic one time, and I wish I had copied it down, but it was something like it takes just fraction of a second for an oxygen molecule to move into our cells because of the size they are. But if our cells were like 10 times bigger, it would take 6 months for the oxygen molecule to diffuse into the mitochondria. I mean, it was something like that drastic of a difference because of the rate of diffusion, because of the speed at which it occurs. And So we have to have cells that are very small in order for transport to happen quickly enough, have it not have to travel very, very far. And we can see that with some of our specialized cells. Do you know, what is the longest cell in your body? Do you know?
Nerve cells?
Very good, your neurons. You have neurons that stretch from your spinal cord, a single neuron, all the way down to your big toe. That's a really long cell, but it's very elongated, very slender, microscopically so, of course, so that there is maximal surface area, so that still that diffusion does not have to occur over a very long distance because there's so much membrane in the way that those cells are formed. What are other adaptations to maximize surface area? Think about our brains. What's true about the human brain that we don't see in some animals that aren't quite as smart as you and I are? But you
All
know,
the
when you look at the
folds
outside,
in the
right,
brain.
very good. It's very folded, okay? Our brains, if they weren't folded, they would be about 1 meter by 1 meter. I mean, we'd be kind of top heavy, right? In the wind, we couldn't fit through the doors, it'd be crazy. But we have all those folds to maximize surface area while minimizing the amount of space that it takes up, okay? When we do our cell size lab next week, you're gonna be creating a cell in which you are going to maximize mass and minimize diffusion time. So you wanna think about some of these adaptations like folding, like not pores, but indentations, et cetera, that are gonna allow you to do that because it's gonna be a contest, and we know we like contests, right?
Yes.
Sixth period's already all over it, already doing research, so be prepared. All right, any questions on that? Okay, so let's then talk about prokaryotic cells. We're gonna go through this very quickly because you actually have this exact diagram in your Unit 1 notes. But just in the very remote chance that you do not remember prokaryotic cell structures, let's revisit them and make sure we are comfortable with them. Okay, first of all, let's start with the structures found in all bacteria. So what is the outermost boundary? Up here it is designated with number 1 that is found in all bacterial cells. Cell wall, right? Come on, guys. With me? Cell wall. And if this is a eubacterium, what is that cell wall composed of? Peptidoglycan. Okay, so number 2 up there, no, number 1 up there, sorry, represents the cell wall. Okay, whereas number 3 is interior to the cell wall, also found in all prokaryotic cells, all cells, as a matter of fact, is cell membrane, very good. It's not numbered up there, but bacterial cells are 3-dimensional, as ours are. So they have cell gel in order to give them their shape. What's that cell gel called? Cytosol, very good. What's number 5? Ribosomes, found in all cells as well. Now, did we talk about the fact that prokaryotic ribosomes are different than our eukaryotic ribosomes? This is an important point, and we're going to come back to it a couple of times. But the fact of the matter is, when we look at the ribosomes in eukaryotic cells versus prokaryotic cells, there's a difference. And one of those differences has to do with size. Prokaryotic ribosomes are described as 70S ribosomes. That 70S is a way of measuring the size of those ribosomes. You and I have 80S ribosomes, as do all eukaryotes. Our ribosomes are larger. It's important for a couple of reasons. One of those reasons is that it allows ribosomes to be a target for antibiotics. We have antibiotics such as tetracycline, for example. Their target is those 70S ribosomes that we see in prokaryotic cells. They're not going to give me very severe side effects as a result of that. All cells, of course, have ribosomes because what do ribosomes do? They make proteins. By the way, I posted your pub quiz grades. Hopefully you had a chance to look at those. I'll get those back to you as soon as I get my makeups done, which should be on Monday. Okay, so ribosomes make proteins. All cells need ribosomes. What about number 4? What's number 4 pointing to up there? We could call that DNA. That's absolutely correct. Well, how else could I refer to that? What's that? Genetic material, I can describe it that way. How else could I describe it? Chromosome, okay, it's a chromosome. Okay, what else is the term we use to describe that? I don't use the term very often, but rather than a true nucleus, we could describe that region as being the nucleoid. So any of those terms are fine. It is important much DNA is there? That's a tangled mess, but how much DNA is there?
One.
One single molecule, single helix or double helix? Double helix, very good. And what is the form of that DNA, given this is a prokaryote? Very good. It is circular as compared to all eukaryotic DNA, which is linear. Okay, and that's important for a couple of reasons. Okay, those are the structures found in all bacteria. So let's look at the adaptations that we might see in bacteria, starting out with number 2. What's number 2 pointing to?
Capsule.
Now the capsule, okay, what is that capsule made of? Does anybody remember? Starts with a P though. Polysaccharide, okay? It's typically a polysaccharide type of capsule. And what is the importance of the capsule? Well, how is that an adaptation to the left?
Protection.
Yeah, it makes it sticky, it protects it from the host immune system. We associate the capsule with pathogenicity in bacteria. Okay, another adaptation we see there is number 7. Most bacteria have that. What is number 7? Flagella. Most bacteria are modal, although not all, certainly. And then lastly, does anybody remember the new term that's used to describe number 6? It changed names. Those are fimbria. Okay, those are fimbria, the little hair-like structures that allow bacteria to attach. Not to be confused with cilia. Okay? Prokaryotic cells do not have cilia. They used to be called villi, but now they're not. So, but these are not cilia. Cilia, by definition, are made from proteins that bacteria do not possess. Okay? Any questions on that? You guys are kind of quiet today. Gotta pep up, yes?
It's Friday.
All the more reason to be excited, not to mention,
We have
where
like-
are we?
We have like 10 episodes.
We are in biology. Another reason to be excited, and it's a beautiful day. There's another reason, right? And you had a short week, didn't you?
Yeah.
Oh my goodness, how could it feel like the longest week in the world? That's nonsense. Oh my goodness, oh my goodness. All right. Well, I can only find reasons to be excited and happy. I don't know about you guys. So, you're up, yes? All right, so let's talk about eukaryotic cell structures. And I want to draw your attention to this slide, a couple of things. First of all, this shows a size comparison. That is a eukaryotic cell. Right there happens to be a white blood cell along with some prokaryotic cells. And of course, what do we notice about the size of a eukaryotic cell? They're larger, and they can be larger because they are more complex. So it allows them to grow to a larger size. It also allows them to become specialized. And that's one of the things that we want to focus on in understanding cell structures, to recognize that while you all memorized cell diagrams that probably look very similar, one teacher to another, one school to another, our cells do not look that similar. They don't look that similar to those diagrams, nor do they resemble each other. We have very different types of cells in our body, and they look very different because of the job that they do. And when we learn about the organelles, we see different predominances of organelles in different cells based on what their job is. Okay? So when we look at eukaryotic cells, okay, we can divide up the parts of the eukaryotic cell. Remember, cytoplasm by definition is the cytosol along with the organelles, so kind of the innards of the cell, so to speak. So we're going to talk about these different components. What I want to draw your attention to is this endomembrane system, because I don't think that terminology is used in Bio 1. And it's important to understand the interconnectedness of this. This allows for very easy transportation and movement between these various structures because they're all surrounded by the same type of membrane. And we can just have these, for example, molecules being enclosed in a piece of membrane and moving from one of these structures to another very easily within the cytoplasm of the cell. All right, so let's see how far we can get today. You're not going to have to label a cell diagram in Bio 2, but we do want to recognize the different types of cells. So in looking at that illustration up there, cell on the left or on the right, which one of those represents the plant cell? Very easily to identify. Oh, the one on the right, correct? And what are some things that give it away as a plant cell?
So.
Okay, cell wall, very obvious. What else? That vacuole, okay, plants we know have a large vacuole, very important to the integrity of that plant cell. And then what's the third means that I have to identify plant cell very easily? The chloroplasts, of course. Have to have some place where we have those pigments to absorb light energy if we had a photosynthesizer. All right, so we're gonna identify the structure that's being described. I expect this will go very easily for you, right? Right, so the control center of a eukaryotic cell, what goes in that top bullet? The nucleus, of course. We said what makes the structure in a eukaryotic cell a nucleus as compared to the nucleoid that we associate with a prokaryotic cell is what? What goes here? Yeah, the nuclear envelope or nuclear membrane, you can describe it as either of those terms. Either is correct. So when we look at that nuclear envelope, how is it different from our cell membranes? What do you recall?
Only RNA gets out, right?
Okay, it is a double phospholipid bilayer. Okay, so rather than just a phospholipid bilayer, we have a double phospholipid bilayer. So what am I going to draw here, a circle or two lines? A circle, yeah, because polar likes polar, right? And so it looks like this all the way around, a double bilayer. That is important because it regulates what comes and goes from that nucleus, and of course that's critically important to the health of the cell because the DNA must be protected at all costs for that cell to survive and be able to maintain homeostasis. But as we know, there are some large things that have to come and go from the nucleus. They cannot get through that double phospholipid bilayer, so how do they travel to and from the nucleus? Through those nuclear pores. Those nuclear pores are simply protein channels that are embedded in that double phospholipid bilayer. And it's important to know what some of the things are that need to travel through nuclear pores. What is one thing we know has to be able to exit the nucleus in order for the cell to make proteins? RNA, right? So RNA has to be able to exit the nucleus. It exits the nucleus via those nuclear pores. What else do we know has to be able to come and go from the nucleus? Well, let's fill in this next bullet. Where are ribosomes produced? In the nucleolus. It's important to understand the nucleolus is not a separate structure. It's just a part of the nucleus that stains more darkly. And the reason that it takes up more stain is because it's a very busy part of the nucleus because it's constantly synthesizing what? If I'm making a ribosome, how do I make that? What are ribosomes composed of? What two components? Do you know?
RNA.
Very good. rRNA, the R standing for? Ribosomal RNA and protein. Very good, okay? So ribosomes, we need to know, made up of rRNA and protein. That rRNA is going to be synthesized in the nucleus from rRNA genes. Where are the proteins coming from? Well, where are proteins made in the cells? The ribosomes, where are the ribosomes located? Yeah, they're out in the cytoplasm, right? So my point is, those proteins are going to be synthesized at the ribosomes. They're going to have to be imported into the nucleus in order to build those ribosomes. And then what has to happen with the ribosomes? They've got to be shipped back out again, correct? Right? Huh? I know, it seems kind of crazy, doesn't it?
Yeah.
But RNA doesn't have any means to get out into the cytoplasm of the cell. So all of that transfer that we just described happens via those nuclear pores. It allows for the proteins to be shipped in, the ribosomes to be shipped out, as well as mRNA to exit the nucleus, as well as tRNA. Yeah? Those words all ring vague bells? Perfect. All right. And then lastly, DNA wrapped in protein. What's the name we use to describe DNA wrapped in protein? Chromatin, okay? And normally, when we use the word chromatin, we're talking about those fine strands of DNA that we see in the cell when the cell is doing its cell job. And oh my gosh, I can't remember, during the cell cycle, is that interphase or M phase mitosis when the cell's doing its cell job? Interphase, very good. So during interphase, which a cell spends at least 90% of its time in, if not more, when it's doing its cell job, we see that DNA fine strands in chromatin form. We used to think that that DNA was just kind of stuffed into the nucleus, just kind of randomly, okay? By the way, you know, you have like 6 feet of DNA in every cell. That's pretty amazing, yeah? Well, we know cells are microscopic. I think it is. But as it turns out, it's not random the way the DNA is located in that cell, not surprisingly. DNA is organized in the nucleus. Each chromosome has kind of an address, a zip code, as to where it's located in the nucleus so that it can be used as it should be. So it can be transcribed and translated. Okay, any questions on the nucleus? What was that, a yes or no?
No.
Good, all right. What goes here? Suspended in cytosol or bound to the rough ER? Ribosomes, okay, so proteins are produced at the ribosomes of the cell. And as we said, ribosomes are described by their location when they are making proteins. If that ribosome is suspended in the cytosol, I describe it as free, okay, it is a free ribosome. What types of proteins would be synthesized by free ribosomes? Proteins that are going to be used in the cytosol. Okay, there are lots of enzymes that are found in the cytosol, as well as other components, cytoskeleton, etc. What type of ribosome would be found on the rough ER? Well, first of all, we described that ribosome as bound. What types of proteins would that ribosome be making? Well, remember the rough ER is part of the endomembrane system. So any protein that's going to be exported out of the cell is going to be synthesized by a bound ribosome. In addition, if that protein's destination is a cell organelle, it's going to be, for example, an enzyme in that organelle, etc., it's going to be synthesized by a bound ribosome. So it can be moved in a vesicle around the cell very readily.
Could you repeat the first part?
Yes, what was the first part?
Makes
Get me
protein-
started.
Sorry, it's like makes proteins where if it's a bound ribosome?
That's either destined for export, or it's going to become part of one of the organelles of the cell.
Oh, thank you.
You're welcome. Okay, any questions on ribosomes? Alrighty. So let's talk about the endomembrane system. Most of that endomembrane system is made up of ER. Although cell diagrams don't show it because we're trying to show a lot of stuff, most of the cell is ER. Okay, it extends all the way through the cytoplasm of the cell. Notice that the ER as phospholipid bilayer is continuous with the outer phospholipid bilayer of the nuclear envelope. So it's just a continuous progression of membrane. We've already basically talked about the rough ER. The job of the rough ER is? Yep, to make proteins that are destined to be shipped out of the cell, or, as I said, to become part of a cell organelle. Of course, we call it rough because those ribosomes make it look rough with an electron microscope. Smooth ER has a lot of jobs, and we call it smooth because
Ribosomes.
there's no ribosomes, okay? So what is the job of the smooth ER? What are some jobs? It detoxifies. That is a word that is always confusing to students, the difference between detoxify and disinfect. Detoxify means to chemically change poisons. Okay, that's what detoxify means. What organ in our body has the job of detoxifying? The liver. And so when we look at liver cells, we would expect to see an abundance of smooth ER, and we do. The other thing that it's important to note in understanding that cells are specialized and look different one cell type to another is that cells are dynamic. They're constantly changing. So if you have a situation with somebody who is a very heavy drinker, what we're going to see in their liver cells is a much greater quantity of smooth ER than someone who is not. Why? Because they are constantly feeding their body poisons that have to be detoxified. And of course, what can happen over time with someone who ingests too much poison on a regular basis, specifically alcohol, but drugs too, is that those cells ultimately cannot handle all the poison that's being fed to them, literally, and they start to die and they scar. And of course, that's what ends up as cirrhosis of the liver. As those cells scar, you cannot live without a liver, scarred cells are non-functional, and ultimately that becomes a fatal disease, most certainly, okay? The other thing that we see with that type of situation is if I'm someone who is a very heavy drinker and I go on the wagon for 6 months and then fall off and take a drink, that drink's gonna affect me much more, because what has happened, my cells have healed. They have gone back to normal amounts of Smoothie R because I haven't been requiring that constant detoxification. So when I go back to my former habit, and you see this happen with drugs sometimes, I know I've had students who had drug problems, they were off the drugs for a while and went back on and took a fatal dose. And it was a dose they had been used to before. Why? Because of the healing that occurred in their cells. They didn't have the Smoothie R network needed for that level of poison anymore. And it became toxic to the cells instead. Okay. What else do we use Smoothie R for? What's another function of it? Guys, there's 4, we're only on 1.
Do something with calcium.
Something with calcium, store calcium, okay? Smoothie R stores calcium ions. So we're gonna see extensive ER, Smoothie R in muscle cells, for example. Muscles cannot contract without calcium. We'll talk about why later. What's another job of the Smoothie R?
Manufactures lipids.
Very good, okay, lipid manufacture. So if my job as a cell is to make what type of hormones do we associate with lipids? Steroid hormones, I'm gonna have a lot of Smoothie R, right? Because that's where that is done. And then the last job of the Smoothie R? Very good, glycogen metabolism. Again, we associate that with liver cells along with muscle cells. So muscle cells, liver cells, extensive Smoothie R. Okay, any questions on the Smoothie R? Alrighty, cell postmaster, what's that? Golgi, okay? The Golgi apparatus, complex, body, it goes by a lot of names. The important part of that name is Golgi because it was Dr. Golgi, literally, who discovered the Golgi apparatus. I call it the cell postmaster. Why? Because it's what receives molecules that are destined for export out of the cell. It basically modifies them, puts them in a package, sticks an address label on them, and ships them out of the cell. So how does that actually work? Well, again, these are going to be molecules synthesized in the endomembrane system. And so as, for example, let's say insulin, as that insulin is being synthesized, it's going to be pinched off in a piece of membrane and make a transport bubble we call a vesicle. That vesicle is going to be moved from the rough ER to the Golgi. Now, what did I tell you yesterday? What are the tracks of the cell? Microtubules. So it's going to be moved along microtubules to the Golgi. The Golgi has sidedness, okay? The part of the Golgi that is directed towards the nucleus, the receiving side, that's called the cis phase. The part of the Golgi that's directed towards the cell membrane, the shipping side, that's called the trans phase. And one of the things that we don't understand right now is when that vesicle arrives at the Golgi, is it moved from sac to sac, which we call cisterna, but you don't need to worry about that vocabulary word. So is it moved from sac to sac, or do the sacs move? Is the whole Golgi dynamic? We don't know for sure, but we know there is that movement as processing occurs. Sorry, my shoe's wrong. That processing may include adding carbohydrates, right? Those types of things are done in the Golgi. Okay. By the way, if that was insulin that we were talking about, as it moved through the Golgi and left via that transphase in a vesicle, it's going to be directed towards the cell membrane and spit out through what process? Exocytosis. Okay. Any questions on that? Marvy. All right. Next thing, how do we identify this? Used for recycling, destroying pathogens, disinfecting. Lysosomes. Lysosomes, we typically say are only found in animal cells, but there are some biologists that feel like there are lysosome-like structures in plant cells. So that's not a hard and fast rule. By the way, Lysol is a product, is named after lysosomes, okay? Because Lysol's job is to disinfect, right? That's why you spray it on things. So again, have to understand, detoxify, chemically change poisons. Disinfect, destroy pathogens. They are two very different words that students get confused because they both mean destroying bad stuff, okay? So lysosomes disinfect, destroy pathogens. What cells would we associate with having a high number of lysosomes? Sure, our white blood cells, absolutely, because that's their job. Another very important job of lysosomes is to recycle. Our cells are much better recyclers than we are as people. They recycle very efficiently. So as biomolecules, enzymes get degraded, et cetera, their parts are recycled and reused again in the cell. And that's done in the lysosomes. So how do lysosomes do that? Well, they contain hydrolytic enzymes. That word hydrolytic should sound familiar, yeah? Was that an emphatic yes?
Yes.
Yes, so what's hydrolysis?
Breaking
Breaking with water, okay? So they break things down with these enzymes, use water to do that. As you can see, we have to be very careful how things are moved to the lysosome. So if a white blood cell takes in, for example, a pathogen, it's gonna take in that pathogen via endocytosis, specifically phagocytosis, and close that pathogen in a vesicle, and that vesicle is going to be moved via microtubules to a lysosome. It's going to fuse with the lysosome, and then that pathogen is going to be destroyed by those enzymes. So the danger with the lysosome is autophagy. What does autophagy mean? Self-eating, right? What would happen if the lysosome in my cell ruptured? Why wouldn't the cell be destroyed by those very same hydrolytic enzymes, right? That's a problem. So there is a mechanism to make sure that does not happen. And that mechanism has to do with pH. What do we know about enzymes with regard to pH? They're pH sensitive, correct? As it turns out, the pH in my lysosomes is significantly lower than my cytoplasmic pH, than the pH of my cytoplasm. What does that mean about my lysosomes? Are there more hydrogen ions or fewer? More. So, how could we end up with more hydrogen ions in lysosomes as compared to the rest of the cell? I bet the answer is in your notes from yesterday. Very good, a proton pump. Remember, we talked about proton pumps embedded in the membrane that ship hydrogen ions in against the high concentration gradient, right? We've already got a higher number in there because we've got a lower pH, so we're moving against the gradient. Those proton pumps, very important. They protect me from my very own lysosomes. Last thing I want to say about lysosomes, how many of you remember learning about Tay-Sachs disease in Biology 1? Does that sound familiar at all?
Yeah.
Terrible genetic disorder inherent to, we used to associate with the Jewish population, we had a higher predominance there. That is one of a large number of genetic disorders called lysosomal storage diseases. And what happens there is that one of the enzymes in the lysosomes, the gene has mutated, the enzyme is not made properly. I ran this in, I look like you're a million miles away. The enzyme is mutated, it's not able to do its job, so with stuff that's supposed to be broken down is not. And as that stuff accumulates in the lysosomes, ultimately it becomes toxic to the cells. Okay, that's what we see with Tay-Sachs. The babies are born normally, about a year, they regress, they learn how to sit up, they crawl and maybe walk, they start to regress. Typically death occurs before they're 2. And it's just because the lysosomes just get chock full of molecules called gangliosides. Yeah. Okay, any questions on those? Okay, last one we're going to talk about, and then we'll let you go back into your pre-lab. What is the term we give to the storage sac of the cell? Vacuole. Now you and I have vacuoles as animals, but our vacuoles are much smaller as compared to the very large central vacuole we associate very characteristically with plant cells. What's the purpose of the central vacuole in a plant cell? Bless you. Provides shape and structure and stores water, okay? That vacuole is very important in the structural integrity of a plant cell. We talked about the fact that we are watering produce continuously, even though they're dead. Why? Well, you tell me. Where do we have the greater water potential if I'm spraying water at those plant cells? Outside the cell or inside the cell? Outside the cell, right? Greater water potential, more solute in the cell. What direction's water going to move? Into that plant cell in order to give that plant cell structural integrity. It also allows plants, they grow not just by making more cells, but plants actually do grow by making bigger cells. How can they make bigger cells? Because of that vacuole. They fill up that vacuole. I'm not increasing the needs of the cell because I'm just making a bigger storage sac of water in the cell. Okay, so it stretches those cells out. Contractile vacuole, why do produce have a contractile vacuole? Because they live in an environment with a high or low water potential.
High water potential.
Greater water potential, right? So water moves into the protist, and the contractile vacuole pumps it back out. Yep? Yes? Okay. All right, guys, you've got about 10 minutes. That's enough time to do